Hello, this is Dr. Jabbour. Today we're going to go over meiosis, and this would be very important to watch after watching the lecture on mitosis, the cell division, um, and the cell cycle. There's a separate lecture about that. So this is also a type of cellular division, but this is very specific to the reproductive organs and to produce sex cells, the gametes. So that's what we're going to talk about in this lecture. So living organisms, um, like human beings, they are distinguished from other structures, other things, by their ability to produce their own kind. This is what's specific to living things. You can uh, reproduce yourself and make more of yourself. That's very characteristic of living organisms. For example, a table is not going to spontaneously duplicate into two tables, and uh, your pens are not going to just multiply. But living organisms do, and uh, that's what sexual life cycles are, and cellular reproduction. I'm going to introduce a few terms here. The term heredity is a transmission of traits from one generation to the next. So if we're going to just stick to talking about humans, we're talking about, transferring characteristics of a person to the next generation. So your mom's curly hair to yourself, or your dad's long legs to yourself. So you inherit certain traits from both parents, and that make you a unique person. Now, what makes you a unique person is that variation. And variation is demonstrated by the differences in appearance that offspring show from parents and siblings. So offsprings are the, the, the term we use in genetics for the children of the two parents that reproduced. And these offsprings are also different from each other. So the children are different from each other. So the siblings are not carrying the same traits or expressing the same traits. And that's what variation is. And it's very important in human reproduction. And in this chapter, we're going to learn about how is this variation introduced, how unique it is and how important it is, because we need to be all different. Otherwise, we would all be clone of each other. All the siblings would be identical and identical to the parents, and it's just not the case. Just think about one thing. If, for example, if we were all clones of each other and we were all carrying exactly the same traits, imagine if we were carrying a trait that would actually not allow us to adapt to a particular condition. But then we would all die, and it would be the end of our species. So being all different from each other allows us to select what traits are actually going to help us survive the conditions that we're living in. And eventually, we're going to evolve into organisms that are very adaptable to our environment. And it's constant, right? So it's over, thousands of years, these traits are like curated for the specific environments. It doesn't happen overnight, but they are selected. These traits are selected so you can survive and keep reproducing and not die and disappear as a species. All right, and the term genetics is the scientific study of heredity and variation and how this all works. So there's a separate chapter on Introduction to Genetics that I will provide separately. This is more specifically about meiosis and what happens, how do we end up with gametes, all different from each other. So look, this is a great picture to show you this variation. Here's a couple, a mixed couple. he has curly hair and darker skin, and she has very straight hair and very blonde hair and fair skin and blue eyes. And here are their children. They are all different from each other. Um, some have lighter hair color, some have darker hair color, some have curly hair, and some have straight hair. It's amazing. So they're all different from each other and different from their parents. So at the end of this chapter, you can understand how that's possible. So the whole story starts here when a sperm fertilizes an oocyte. So here's the sperm, and he is with millions of other sperms, and they're all swimming towards that oocyte. I call it an oocyte. This is the proper term when we talk about female reproductive system, but in a lot of textbooks you'll also see the word egg. So the sperm is going to go fertilize the egg, but the sperm is competing with so many other sperms. And who's going to win the race is the sperm that's going to be the fastest, the sperm that has the best set of enzymes to help it get through the protective layer of the egg, because there's two protective layers over the oocyte itself. Oocyte is just a cell, so the egg is probably the cell with the coverings over it. So you have to have a sperm that's going to be able to get through these coverings and swim fast, and also avoid everything in the female reproductive tract that's going to, prevent it from advancing, because the female reproductive tract is helping select the best one, so it's making it hard. So the weak ones are left behind. So the sperm, when it reaches the oocyte, when it reaches the egg, it's going to, dig a tunnel. Okay, but once it reaches the surface of the oocyte and it touches the plasma membrane, there is a whole chemical reaction that takes place, and you'll learn that in the female reproductive system chapter. But that's then and only then that you have the DNA. Let's do it in blue so it stands out. The DNA, which was packaged in the nucleus over here, the DNA is going to get injected in the oocyte. The entire sperm cell actually stays on the surface of the cell membrane, and really the only thing that comes out is the genetic material. So it's literally an injection of just the DNA. So you have this one cell, and you can see this is a huge cell, right? This is one big cell. This is going to be your first human cell of this new human being. And it's basically made of that big oocyte that has a lot of cytoplasm in it, and the DNA of the sperm cell. Of course, this oocyte has its own DNA. So I'm going to draw it here. The DNA that was already in here, and they're both going to fuse together to form this new DNA set that basically includes, I'm writing here 2N. I'm going to explain to you what that means in a second. It's going to include the set of chromosomes from mom and the set of chromosomes from dad. So the set of chromosomes that were in the oocyte and the set of chromosomes that just got injected by the sperm cell into that oocyte. All right. So your first human cell is the original oocyte plus two sets of chromosomes, one that was already there from mom and the one that just got injected from dad's sperm. All right. So this offspring, this new cell, it's going to be basically now a new human being, acquires the genes from parents by inheriting these chromosomes. So I'm going to take the next page to draw these chromosomes and just remind you of what we have here. There we go. So, and I used pink and blue, I guess, yes. So the sperm is going to provide a set of chromosome 1, chromosome 2, chromosome 3, etc. And the 23rd kind of chromosome is the sex chromosome. And it's not called chromosome number 23. It's actually called a sex chromosome, and it's going to be either X, or Y. It's one or the other. Okay, so this is the set of chromosomes injected from the sperm. All right, and in pink, I had done the chromosomes in the oocyte. So in the oocyte, we already have a chromosome 1, we have a chromosome 2, and we have a chromosome 3, and the only possible sex chromosome that we have in an oocyte is an X chromosome. So the pair of sex chromosomes in this new fertilized egg, it's going to be either XX, or if the sperm that won the race and arrived to the oocyte and injected its DNA happens to have a Y chromosome as its 23rd chromosome, then we will have a XY pair of sex chromosomes. So it's either one or the other. It's either this or this. It can't be both. All right. So this is a pair of chromosome number 1. This is a pair of chromosome number 2. And this is a pair of chromosome number 3. And number 4, number 5, number 6, 10, 15, 22. And this is the 23rd pair of chromosomes. So it's number 23rd, right? I said that. So it's the 23rd type of chromosome that comes in a pair, either XX or XY. Okay, so you can appreciate now why it's important for each gamete, by the way, this is the word we use here, a sperm is a male gamete, or sex cell, and the oocyte is a female gamete or sex cell. Okay, so make sure you know these terms. These are important terms. So these gametes, they need to have a set of chromosome 1, 2, 3, 4, 5 till 23, because when they come together, and this is something I would have asked you in class if we were together in a live lecture, hey, what do you think? Why do you think we should only get one set of chromosomes in each one of these gametes? And I would have given you some time to think about this, and you could pause this video and think about this before you hear me say why. It's because when they join together, they're going to form the pairs together. So if they started as holding pairs of chromosomes already in them, in these cells, if we already had pairs of chromosomes, then we would have pairs of chromosomes in the sperm cell, pairs of chromosomes in the oocyte. When the sperm injects its DNA, now we're going to have, a pair of each chromosome 1, and the oocyte is already providing a pair of chromosome 1. So now we have 4 chromosome 1s. That's too many. So we're going to end up with 90, so 23, 46 chromosomes. If they were holding 46 chromosomes each, we would end up with 96 chromosomes in the first fertilized egg. No, that's just too many. Our cells, all the cells in our body, are 2n. That means they have each chromosome in a pair. And n is the number of chromosome types. So organisms have chromosomes in pairs, most organisms, eukaryotic humans. And for humans, number of chromosome types is 23 for humans. Now, for dogs, it's a different number. For elephants, it's a different number. And I don't have these memorized. So I don't know how many chromosome types the other animals have. I didn't I pull out this information before starting this recording. But humans, we have 23 types of chromosomes. All right. So they come in a pair in all of the cells in our body. Our cheek cells, our muscle cells, our neurons, every cell in the body has 2N chromosomes, has 46 chromosomes. And they look like that. They're in a pair, like this. Pair of chromosome 1, pair of chromosome 2. Right. So these are called, all of these, till number 22 here. These are called autosomes. And the 23rd pair is called the pair of sex chromosomes. Okay, so autosomes and sex chromosomes. Okay, so all of our cells have that. But if we have sperm cells and oocytes that have pairs of chromosomes, when they eventually meet, it's going to be too many. So we need to reduce the number of chromosomes by reducing the pairs to being only one of each type. All right. So in order to have pairs of chromosomes in all of our cells, the gametes need to provide a set each. So in the end, we have a duplicate of each chromosome type. Also, let me remind you what these chromosomes have in common, or how they are different from each other. What they have in common is that they're all holding genetic information. Now, what's different is that they're all holding different genetic information, different genes. So I'm going to use a slightly different thickness of pen. There's a gene here that would code for something, and another gene here, and another gene here. And actually, I really should have done something a little bit different. Here's green, and here's light green. And here is dark blue, and here is light blue. And I'm going to explain to you why in a second. Here's orange, and here is, oops, where did it go? There we go. So why I actually picked them slightly different colors, a slightly different shade of the same color, it's because that's how it is in reality. Right here, this is called the locus, which is basically the location of a gene. So here's another gene at a different locus, and here's another gene at yet a different locus. So these are genes. Right here, this is a gene, and this is the other variation of the same gene. So what that means is you have two different ways of making the same thing. And my analogy, which is really going to be helpful to understand this, is imagine all of these chromosome types being different cookbooks from different countries. So let's say chromosome 1 is holding all the recipes to make up dishes from, let's say, Italy, and chromosome 2 is holding the recipes to make up dishes from Lebanon, and chromosome 3 is holding up the recipes to make up dishes from France. So those would be completely different dishes in different cookbooks. You're not going to find a Lebanese dish that's going to be described in a French cookbook. So these are different types of chromosomes because they're holding different codes, different recipes to make these different things. All right, so now when you are looking at, let's say, this Italian cookbook, on page, so the locus is the equivalent of a page number. on specific page numbers in that cookbook, you would have a specific recipe. And that's what a gene is. The gene is the recipe to make whatever that product is that you want to make. So let's say we want to make meatballs. So the meatball recipe is on page 732 in that cookbook number 1, because that's the Italian cookbook. So when you're looking at mom's cookbook and dad's cookbook, same page, same recipe, there are some variations between the recipes, because even though in the end we really are going to get meatballs, maybe dad adds more spices to it, mom adds more basil to it. In the end, it's still going to be meatballs, spaghetti meatball dish, Italian dish from an Italian cookbook. And that's what it is. And that's the difference between these the same gene can have different alleles. Alleles, allele of a gene. So an allele is a variation of that same gene. So mom's allele and dad's allele code for the same gene, but with a few little differences in the code, that it will make a slightly different version of that product that you're making in the end. So very simple example, just very simple. For example, if you your body needed to make more keratin, which is the protein in your hair, maybe because following chemotherapy, your hair is regrowing, so it's rebuilding, hair shafts, and keratin is needed. So we need to make keratin. So the gene to make keratin is on a specific chromosome, and I don't know which chromosome it is on in actuality. But dad's version of keratin is straight, while mom's version of keratin is more of a loopy molecule. So your hair will end up looking more curly than if it was just coated with dad's allele. So that's what it is. We inherit traits that are going to be different from dad and mom, and we are a combination of these traits. Okay. Did I say everything I want to say here? Okay. So this is what we call diploid cells. Diploid cells in our body have a 2n number of chromosomes. So 2n is equal 46 in humans, because n is equal 23 in humans. So because every chromosome type comes in a pair, we end up actually having 46 chromosomes in all of our cells. All of our cells in our body, we have 46 chromosomes, except the sex cells. So the sex cells, they start with 46 chromosomes, but eventually we need them to have 23 chromosomes. We need them to split the pairs from each other. So then we have sex cells that only have 23 chromosomes. So when they are paired with, the sex cell from the opposite sex, it also has 23 chromosomes. And when they come together during fertilization, we will end up creating the first cell with 46 chromosomes. And that's it. That's your first human cell. The fertilized egg has 46 chromosomes, the 23 that came from mom and the 23 that came from dad. And we have our first diploid cell. Diploid cell means it has pairs of chromosomes, okay? So a sex cell that has half the amount of chromosomes is called haploid. Haploid cells do not have pairs of chromosomes. They only have one of chromosome 1, one chromosome 2, one chromosome 3, one chromosome 4. They only have one or the other. And we call them haploid cells. And the only haploid cells in your body are the gametes. That's it. Everywhere else in the body are diploid cells, and the only haploid cells in your body are the gametes, because those are the ones that need to have half the amount of chromosomes. So when they eventually meet for reproduction, it will be the first human cell with 46 chromosomes. Okay, So I think we covered that enough. And now let's see, how do we create these haploid cells? That's the purpose of this chapter, is to see how do we create a haploid gamete. All right. So this is going to be kind of repeating some of the things that I already drew. Genes are the units of heredity and are made up of segments of DNA, which is what I drew here. Boom, boom, boom. Genes are passed to the next generation via reproductive cells called the gametes, or the sex cells, sperm and eggs. Most DNA is packaged into chromosomes. Um, uh, so you will hear the words used interchangeably, chromosomes, DNA, but technically speaking, DNA is a nucleic acid that looks like this, which is a double helix. And it has histones, proteins that are attached to it. And the histones, they help coil the DNA. It actually, you'll see in the animation, the histones help coil the DNA. And when the DNA is all coiled around the histones, it creates these little tight chromosomes. And that's what we want when we're starting to divide the cell. We want this chromosome to be tight. So the chromosome, technically speaking, is DNA plus histones. Humans have 46 chromosomes in their somatic cells. So we already saw these terms. Oh, no, I forgot to tell you. Um, So diploid cells, let me make some room here. Diploid cells, all the somatic cells in your body. These are all the cells in your body except the sex cells. So, and who is haploid? The sex cells or gametes. So sex cells are haploid, somatic cells are diploid. Somatic cells, all the cells of the body except gametes and their precursors. Precursors because this is a diploid cell, right? So this is the precursor of the gametes. So the precursor has 46 chromosomes, is a diploid cell, and it goes through a series of cellular division to create haploid cells. And now we have gametes. A gene-specific position along chromosome is called a locus. I already mentioned that in the drawing. All right, so let's just quickly look at asexual and sexual reproduction. In asexual reproduction, a single individual passes all of its genes to its offspring without the fusion of gametes. So it's basically one cell that's going to divide into two cells, and these two cells become the parent cell and divide into two more cells. It's kind of like what bacteria do. They just basically split themselves. And a clone is a group of genetically identical individuals from the same parent. So one cell giving rise to two cells, so that usually we call this the parent cell, and it gives rise to two daughter cells. These two daughter cells are going to give rise to two more daughter cells, etc. And this is really how bacteria grow, by this type of reproduction. And all the cells are exactly identical to each other. And that becomes a clone. So if you took microbiology, you will learn about cell colonies, where all the cells are kind of on a mound like this on the plate where they grow. And basically that colony is all the cells that divided, and they're all basically coming from the same original cell, but they grew, and over, 1 day or 2 days, and you come back and you see there's like these little colonies on your plate. You realize this is a mound of cell that originated from one cell, the original cell that landed on the plate originally the day you plated the cells. One cell gave rise to all of these cells on top of it, and that created a colony. So, no, really wasn't necessary to go into that kind of detail, but that's okay. In sexual reproduction, you have two parents that give rise to an offspring, and that's a very unique combination of genes that is inherited from the two parents because the gametes they're providing to fuse together, they are very different from each other, and the choices of gametes are varied, so you're going to end up with a very specific and unique individual in the end. So very different. This is also something I already said. Human somatic cells have 23 pairs of chromosomes, and that's why somatic cells are diploid. So I'm just reminding you of that. This is what this means, pairs. So we write the number 2 here, this to represent that these are coming in pairs. Okay, so all these cells are diploid cells. Now, what do we call a karyotype? It's basically a picture of all these chromosomes, all these 23 types of chromosomes, and finding their homologs. So that's a word I haven't mentioned in my drawing, so I'm going to add that here. These chromosomes are described as homologs. 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 We also say homologous chromosomes. So homologs mean that they are the same kind. It's both are chromosome number 3, but they're homolog, meaning they're not exactly identical to each other. Otherwise, we would say identical. But we don't say identical because they're not identical. They're homologs. They're similar. Homologs means similar because it's the same chromosome type, but there's variation between the genes. And if you remember, I told you that for every gene, there are several alleles, and mom and dad can actually be carrying different alleles for the same gene. So it will be a slightly variation of the product that's going to be coded by that gene. Homologs. So chromosomes in a homologous pair are the same length and shape and carry genes controlling the same inherited characters. But there could be variation between these two genes. The two codes for the same gene, not two separate genes. So the two codes for the same gene. And that's why we use the term homologous. So you basically create a smear on a slide, and you spread out these chromosomes, and they're very condensed, so they're very tight, and you can actually see them separated from each other, and can literally go and look for which ones look the same and arrange them by basically taking pictures of, this one and this one. Oh, I think those are the two the same. And this one and this one. Oh, those are the two the same. And this one and this one. And what the scientist does is it's making sure that this particular sets of chromosomes is actually carrying the right amount of chromosomes. So I'm going to show you a picture of a karyotype. So this is what a karyotype is. This is a karyotype, which is basically the picture of all 23 pairs of chromosomes in a human being. So look at chromosome 1. It's very long. It looks like to be the longest chromosome of all. Chromosome number 1. And here is its homolog. So these are homologous chromosomes, chromosome number 1. These are homologous chromosomes, chromosome number 2. Homologous chromosome, chromosome number 3. So there were specific staining that was done on these chromosomes. So we are able to actually identify the pairs that correspond to each other because of the coloring that ends up being on these chromosomes. And we're like, oh, these two have exactly the same order of pink, yellow, dark green, light green, green, yellow. And so they're both the same chromosome, even though this one might be a little bit more bent, on the slide, but those two are chromosome number 2. And these two are chromosome number 3 from the way that they look. So visibly, the scientist has to arrange them in pairs for the reason to see, is everybody looking okay? And we also can see, for example, 19, 20, 21, 22, and here is the 23rd pair. We don't call it 23rd. You see, there's no 23 here, but this particular 23rd happens to have one X chromosome and one Y chromosome. And it's the only pair that is not considered a true homologous pair of chromosomes, because X and Y are actually completely different chromosomes. And this would be for a male organism, a male human being, XY. So XY is for boys, and XX would have been for a girl. But here it's an XY. So for XX, it would be considered a pair of homologous chromosomes because both are X. But if it's a boy and we end up having an X and a Y, that's the one exception of using the term homologous, because they are not homologous. The Y chromosome contains very specific sets of genes that drive the development of an embryo into a male fetus. So in the beginning, the embryo is technically like a female setup, and because of the presence of the Y chromosome, certain things are going to evolve into specific male structures. And if there was no Y chromosome, they would stay the way they are. So there's very specific example that, well, I'm not going to go into that now. You'll see when we cover the reproductive system and embryo development. All right. What else I wanted to say about this? Okay, so the other reason of creating a karyotype is we want to check if all the chromosomes are okay. Is there a chromosome that's missing a piece? That would be really bad, because then, yes, we have 2 chromosomes, and a lot of the times, if one chromosome is defective, the other chromosome compensates for it. But it's not always the case, and it's not all along the chromosome. So sometimes if one chromosome, let's say here on chromosome number 4, I have a mutation right here. And on this particular gene, there's a mutation where the gene is actually not functional. Most of the time, the gene that's on the homologous chromosome, this is going to be okay. And this will produce the product that we need to have. And we are not relying on the one that is not okay. So this is the mutated one. We can't make the product, but thankfully we have the other gene on the homologous chromosome, and we're going to be, using that gene to produce the product that we're interested in. But but sometimes there's a whole piece missing, and sometimes that the missing of that piece is completely detrimental. Also, we look for abnormal number of chromosomes, because the gametes can be carrying an off number of chromosomes. So instead of having 23, the sperm 23, the egg 23, and now we're going to have 46 by combining the chromosomes in pairs. Sometimes a sperm is carrying 24 chromosomes and an egg is carrying 22 chromosomes, so one missing or one extra, because of a mishap during cellular division, preparing that gamete. And the best example is chromosome number 21. Sometimes we have chromosome number 21 that comes in triplicate. So that's what karyotypes are used for, is to just check, do we have too many chromosomes? Do we have not enough chromosomes? Because number 19 is only one and there's no homologous chromosome in there, most of the time, having an abnormal number of chromosomes is actually non-viable. The embryo is going to spontaneously abort. You're not going to have a viable embryo, then fetus. It's not going to grow. And that's how nature takes care of itself, because if the chromosomes are an abnormal number, we wouldn't be able to form a normal human being, and it's just done, right? It's not feasible. So that's how the body has a way of dealing with that. So abortion, not abortions, spontaneous, yeah, spontaneous abortions are our natural way of, getting rid of an embryo that is not going to evolve into something viable. But sometimes there's an abnormal number of chromosomes and it is viable. And for example, here, having three chromosome number 21 is called trisomy 21. Trisomy means 3 sets of number 21. And that's, maybe you already know, that's Down syndrome. So an individual with Down syndrome actually has 3 chromosome 21. All the other chromosome pairs are fine, but this person has 3 chromosome 21 in every single cell of their body. 3 chromosome 21. And they're viable. They grow, they are born, they grow as young adults, that a lot of them are very independent, but there's some mental retardation, there's some fertility issues. So there's some issues. But I hear they're very happy, also happy people. So there is, drawbacks, of course, of having an abnormal number of chromosomes. The sex chromosomes determines the sex of the individual, called X and Y. Human females have homologous pair of X, human males have X and Y. The remaining 22 pairs are called autosomes. We already talked about all of that. Each pair of homologous chromosomes includes 1 chromosome from each parent. I already talked about that. I already talked about all of that. Okay. All right. So a cell is going to, oh, I can't believe I haven't even started talking about meiosis. But really, everything I mentioned so far is very foundational, is very important. So it's not a waste of time to hear this. Maybe this is a review from, I mentioned some of these things in my mitosis lecture, but it's really important, and students have a hard time grasping these concepts, so it's okay. So before every cellular division, the precursor cell that's about to divide always doubles up on everything. Always. We saw that in the cell cycle. In the cell cycle, right before cellular division, there was the G1 phase, S, and then G2 phase. Remember that? G1, S, G2, and we saw mitosis happening during that cell division time frame. Okay, and this is the growth phase. This is the interphase, and it's the purpose of it is as a growth phase. So G, like growth. It's actually meant to say gap, but to remember it, growth, and S is for synthesis of DNA. This is where you have DNA replication. So doubling up on the DNA. So for every chromosome in a pair, you're going to have replication of that chromosome. That chromosome is going to get replicated. And this is a pair of, we don't say homologous chromosomes anymore, but they are homologous. We say a pair of replicated chromosomes. 1, because they're still in a pair, and 2, because they're replicated. The cell just went through the S phase of the interphase, and it's about to go through cellular division. So at that point, all the DNA is also doubled up. So in our case for meiosis, we're like, okay, why are we doubling up on this DNA? Because the intention is to split them and have, only half. So why are we doubling up? Because that's the process before any cellular division. There's doubling up, which means that when we go into cellular division for meiosis, we're going to need to divide twice in a row. After we divide once, we're going to end up really only, technically speaking, back to the number of chromosomes that the original parent had, but we wanted haploid, so we need to do a second cellular division. So meiosis will have two cellular division in a row, as opposed to mitosis. Mitosis, it was just one. Okay, so every chromosome goes through replication, and we end up with, in this drawing only 3 pairs. So the pair of chromosome, maybe we'll say this is chromosome 1, and this is chromosome 1, and this is chromosome 2, and this is chromosome 2, and chromosome 3, and chromosome 3. You see how they're homologous to each other, right? This red chromosome is not the homologous of this one, obviously, because this one's so much shorter, this one's so much longer, so you can visually tell here. But perfect, so we have pairs of replicated chromosomes. This is replicated, this is a replicated chromosome, and this is a replicated chromosome right here, right here. So let me go back to this. This is replicated, this is replicated. Replicated chromosome, and this is a pair of replicated chromosomes. Okay, so the replicated chromosomes, we also call them sister chromatids. Sister chromatids. We call them sister chromatids. So we don't call each individual line a chromosome anymore. We call them chromatids. And as long as they're kind of like the 2 replicated chromosomes are stuck together, we're calling them sister chromatids. That's it. That's the terminology we use. Until we split them apart, we will call them back chromosomes, but right now they're together and they're called sister chromatids. So here you can see sister chromatids of 1 duplicated or replicated chromosome. And so again, this is sister chromatids, sister chromatids. Here you have 2 non-sister chromatids in a homologous pair. So this is the sister chromatid of this one. You see, I mean, I'm saying this and this. Hopefully you're watching me. This is the sister chromatid of this chromatid. And this is the sister chromatid of this chromatid, right? The sister chromatids are of the same chromosome because that's how they ended up being created. It's that one chromosome that gets copied. So there is no sister chromatids between the homologous. Make sure you understand that. Okay. And then you have a pair of homologous chromosomes or pair of replicated chromosomes. So a gamete or sperm contains a single set of chromosomes and it's called haploid. We already saw that. Okay. We already talked about all of that. In an unfertilized egg, the sex chromosome can only be X because that's coming from the mom, and the mom is female, so she will never carry a Y. So all her eggs will have X, 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 X. That's it, X, 1X. But a sperm actually will carry either an X or a Y. So for every, sexual intercourse, there's a bunch of sperms that are going to be dumped, and half of them will be carrying the X chromosome and half of them will be carrying the Y chromosome. So at fertilization, we'll have the union of these gametes with the sperm injecting its DNA into the egg. And that's it. The sperm cell itself will stay on the surface and will, eventually just go away. And it's just the DNA that gets injected in there, and all the cooking will take place in the egg. So when you do have a fertilized egg, it's called a zygote. You definitely have to know this. All the terms in this chapter are important. So after fertilization, you have a zygote. That's the first, well, what am I going to say? I would play hangman if we were in a classroom with all of you in front of me and a whiteboard and behind me. And I would say, what do you think I'm going to say here? This is the first what? This zygote, this one cell. It's what? Diploid. We would be playing hangman in the class, and I would start hanging your little man if you weren't uh, guessing the correct letters. Um, but we can't have fun like that in a recorded lecture, but that's okay. So this is the first diploid cell in a human being. And after that, you have a number of mitotic events that take place because from one cell, you need to have, 2 cells and then 4 cells. And do you know that at the end of the first week, so eventually all of this is mitosis. So here you have fertilization, right? I'm just going to put an F with a little heart. Fertilization, the sperm comes in, injects its DNA into the egg. You have your first zygote. And then the zygote, by mitosis, is going to really end up creating a ball of cells. And this zygote, by the way, is 46 chromosomes, has 46 chromosomes, and it's called diploid. These are haploid cells. This is your first diploid cell. So this ball of cell, did you know that this ball of cell is still not implanted in the uterus? It takes a whole week to get from the fallopian tubes in the female reproductive tract, where the egg was ovulated. It goes into the fallopian tube, and the sperm meets the egg in that tube. And fertilization takes place in the tube. And this fertilized zygote travels down the tube and into the uterus and implants itself in the wall of the uterus, usually in the back wall. It implants itself in the back wall of the uterus and starts burying itself underneath the uterine lining. That takes a week. So by day 7 days after sexual intercourse, if there's fertilization, you will have an implanted embryo. And those 7 days, we're still 7 days away from the end of a female cycle. And it's usually at the end of the female cycle that the woman misses her period and starts thinking, oh, am I pregnant? And, maybe it'll take her a week to kind of convince herself to go take a pregnancy test. And a week later, she takes the pregnancy test. Now we are 3 weeks from fertilization and 2 weeks from that embryo being implanted. So that embryo is already in the uterine wall and growing now. Okay. All right, so we talked about the gametes being produced. Now, when are they being produced? Throughout the entire life of a male or female human being? No. Only at sexual maturity. The ovaries and testes produce haploid gametes. Now, it's a little bit more sophisticated in women because technically speaking, female babies are born with a set of oocytes that are almost ready to be used, and then they will get triggered to be maturing during puberty. In males, it's literally at puberty that we start rolling the production of gametes. But in females, it's actually when she's an embryo that this gamete formation starts. So that's a little bit more complicated story that you will hear when you go through the reproductive system in A and P. So at puberty, boys start making sperm cells, and young women start making mature eggs, and will start ovulating. So ovulation starts at puberty, sperm cell production starts at puberty. Now, sexual reproduction doesn't start right away, but eventually when, a couple meets and decides to have children, fertilization will take place. So this production of gametes takes place in the ovaries in the female and the testes in male. And the process is called meiosis, and meiosis results in one set of chromosomes in each gamete. That's the whole purpose. And fertilization and meiosis alternate in sexual life cycles to maintain chromosome number. So we will, actually, there's a diagram that's going to show that to you. So reminding you here, gametes are the only haploid cells in animals. That's it. Only the gametes are haploids. Every other cell in your body is diploid. They're produced by meiosis, which means meiosis is only taking place in the reproductive system. And once those gametes are made, that's it. There is no more dividing sperm cells even more. That's it. Once they're made, and then it's a stock that we need to use to create a new individual. So then gametes fuse to form a diploid zygote that divides by mitosis. Then you have the mitotic process to end up having a multicellular organisms. So so this is how it alternates. There's a time when we're making the sperm and the oocytes, and there's a time when we're actually doing sexual reproduction for fertilization purposes to create a new individual that will then eventually produce its own sperm or oocytes, and the cycle continues. So I forgot why I put that particular slide here. It's just basically showing the same thing. We're just basically going from haploid cells, which are the gametes, to diploid cells, which are basically all the cells in the human being, in the organism. So just don't worry too much about this slide for now, or at all. Okay, so meiosis reduces the number of chromosomes from diploid to haploid. Oh, did we say this enough? I think we said this enough. So like mitosis, meiosis will be preceded by the replication of chromosomes. I had mentioned that before any cellular division, we double up on everything. So we need to have two consecutive, I'm not used to these. Two consecutive cell divisions called meiosis 1 and meiosis 2. The two cell divisions result in 4 daughter cells. So unlike mitosis, where one parent gives rise to 2 daughter cells, in meiosis, you have one cellular division, you have 2 daughter cells, but then you have to go through a second cellular division process. So now these 2 daughter cells are giving rise to 2 daughter cells of their own. So in the end, you end up with 4. But what's the purpose? What's the purpose? This is the purpose. You have to remember that. Each daughter cell has only half as many chromosomes as the parent cell. Very, very important. That's the goal. Remember, sperm needs to bring 23, and oocyte has 23. And when they fuse, we will have 46. So chromosomes are going to duplicate during interphase. All right. And sister chromatids are very closely associated together along their whole length. And we already said this is called sister chromatids. They are stuck together by cohesive proteins. So here I'm drawing sister chromatids. I'm purposely not drawing lines here so it doesn't look like a DNA helix, because the DNA helix is, technically speaking, it's here, right? This is one chromosome, And this is the other chromosome, right? If we're technically speaking, this is it. But what's linking them together are these cohesins, right? So I didn't want to do bars like that, holding them together, so you don't confuse that with the bars that are within the helix. Okay. Okay. Cohesins, cohesive proteins that are holding these sister chromatids together. Okay. So you're going to have one chromosome of the pair like that, and the other chromosome of the pair like that as well. So remember, each one is a chromosome. And you have the same setup here. The sister chromatids are being held by these cohesive proteins. Okay. All right, so we start with a pair of homologous chromosomes and then they become replicated. So here's your pair of homologous chromosomes. And these are your replicated chromatids, sister chromatids. So on the exam, these are the typical test questions, showing you diagrams of this and asking you, hey, what is this called? What is this called? I want to make sure to know that you know what's the pair, where are the sister chromatids, etc. And actually, they're held at it's not always at the center. They're held together at their centromere, and they're glued together with these cohesive proteins. Okay, so this is the big picture, and this is what you really need to understand. This is the big picture. If you get this, all the details we're going to see coming up are going to be more digestible if you understand the big picture. So the idea is we need to separate the pairs from each other. Boom. Because if we end up not having pairs, we end up being in a haploid stage. That's what it is. We had pairs to begin with, and pairs of one, pair of chromosome 2, pair of chromosome 3, pair of chromosome 4, blah, blah, blah. That was 46 chromosomes total. That's too many for a gamete. So what do? We're going to separate the pairs. That way, each cell is going to receive one of each kind. So it's not going to be missing a chromosome type. It's just not going to get them in pairs. So chromosome 1 of the pair will go to one cell, and the other chromosome 1 of that original pair will go to the other cell. Chromosome 1, chromosome 1. Each daughter cell will get a chromosome 1. Chromosome 2, chromosome 2. Chromosome 3, chromosome 3. So in the end, each one of these daughter cells here, these two daughter cells, will have an entire set of chromosomes. 1, 2, 3, 4, 5, all the way to 23. They're just not going to be in a pair anymore. So homologous chromosomes separate. The pairs separate. The pairs separate. No more pairs. But we still have sister chromatids. And technically speaking, that's one chromosome and that's another chromosome. That's just too many DNA copies. So, and this is really not how a parent cell looks like. A parent cell doesn't have replicated chromosomes. A parent cell has just one chromosome. That's what we need for that final cell. We need just that original chromosome. So this is the original chromosome 1. What happens in meiosis 2, we have separation, splitting of the sister chromatids, just like we did during mitosis. So both of these daughter cells receive the same chromosome 1, and that's what happens in meiosis 2. Every chromosome that's in a replicated form sees its sister chromatids separate, and then they'll be called chromosomes again. That's the big picture. So when we are at the end of meiosis one, we are, technically speaking, in a haploid stage. This is another kind of typical question on a test or quiz. You know, when is the haploid stage technically reached? Generated, like when do we reach this haploid stage? Technically speaking, at the end of meiosis one, because the pairs got separated. That's the whole point here. Originally, the precursor cell had 2N, and then here these two daughter cells only have N. They only have an N number of chromosome types: chromosome 1, chromosome 2, chromosome 3, chromosome 4, chromosome 5, only one of each chromosome type. But they're still in a replicated form, so technically there's still too many copies. So we split those in the second round of cellular division. This is the entirety of the stages, and meiosis one, meiosis two, they follow exactly the stages of mitosis. So we're going to have a prophase, where the nucleus is going to kind of disintegrate, and now the chromosomes are free to, like, line up. Prophase, and then metaphase, they line up at the equator of the cell. Anaphase is when the separation takes place. Telophase is when we wrap things up, and we have two separate daughter cells at the end of telophase with cytokinesis, the splitting of the cytoplasm. So this is followed immediately by the second round of meiosis, which is very similar. Again, we enter immediately. Basically, telophase doesn't actually really finish for each one of these daughter cells. Telophase doesn't really finish. We enter another prophase. We enter a new cycle, a new cell division: prophase, metaphase, anaphase, telophase. So the division in meiosis one occurs in these four phases: prophase one, metaphase one, anaphase one, telophase one, and cytokinesis. So we have to talk about these phases because there's special things going on in prophase one that is not happening in mitosis. You have to know that. You have to understand that. All right, so let's see what's going on in prophase one. In early prophase one, each chromosome pairs with its homologue and crossing over takes place. This is something that does not take place in mitosis. It does not take place in mitosis. So these, they're going to look like X-shaped regions. You'll see I'm going to draw one. Those regions where they kind of like cross over, it's called a point of chiasmata. And this is where the crossing over is going to take place. So I'm going to draw a replicated chromosome, and I'm going to draw its homologue. See, so these are sister chromatids, and this is the pair of replicated chromosomes. I'm not writing everything again. Pair of replicated chromosomes, or pair of homologous chromosomes, but they're in a replicated form. So these chromosomes, what's happening in prophase one? They're like, oh, did you hear? We're going through meiosis. Oh no, meiosis, this is when they separate pairs from each other and we're not going to be together anymore. And they're very, very sad. And this is the time where they're going to exchange souvenirs from each other. They're going to say goodbyes, and they know they're going to be separated soon. These chromosome pairs are not going to go to the same cells. So we have points of chiasmata that are being formed right here. Point of chiasma or chiasmata, where crossing over takes place. crossing over takes place. What's that? What is happening? Imagine you're sitting next to your friend in class, and then you lock up arms with each other, and uh, you're like, we're going to be separated and we're not going to be together in the same classroom anymore. I need a souvenir from you. I'm going to give you a souvenir from me. Click, click. You dislocate your elbow joint and you give your forearm to your friend, and your friend gives you their forearm to you. And now you have, yes, I know this is a crazy analogy, but hey, it works. You have your forearm from somebody else. So now you're really not looking quite as much as like when you were created. So here's at the end of crossing over, this is what things would look like. You would end up with replicated chromosomes that have basically exchanged a piece of their DNA, oops, right here. So eventually, when these chromatids are going to be separated, every single one of them will be different. Every single one of them. Because this sister chromatid will be entirely the original one parent sequence. This green one over here would be exactly like the original chromosome in the parental sequence. So different from, right? The green one here is different from the purple one. And this purple one now has a piece of green, and this green one has a piece of purple. So completely different from each other here and from the sister chromatids within themselves here. These sister chromatids are not really identical anymore. Sister chromatids, by the way, are identical, because this is a replication of the actual DNA molecule, and it's an exact copy. And this is beautiful because this crossing over introduces variety in these chromosomes. So instead of having entirely purple chromosomes or entirely green chromosomes, now we have purple and green. And depending on what we kind of swapped, we're going to be able to do certain things or not in the future person. And we'll get to that as well. All right, so all this drama happened in prophase 1. I want a piece of you, and we're exchanging pieces of like souvenirs, like a piece of the chromosome, swapping pieces. Okay, in metaphase 1, the pairs line up at the metaphase. They do line up at the metaphase plate, with one chromosome facing each pole. So now, unlike mitosis, where the microtubules were coming from both sides, and there was a tug of war between the two ends, what we have here are microtubule fibers that are landing on one chromosome, but not the other. One chromosome, but not the other. And each pole is going to shoot for a specific chromosome of the pair, but not the other. That way, in the end, each pole is going to pull towards it one of the chromosome of the original pair. So microtubules from one pole are attached to the kinetochore of one chromosome of each tetrad. And this is something I forgot to mention, the word tetrad. Well, I'm going to draw these chromosomes here. So let me draw. I'm going to draw them laying down now. Okay, so this was my pair of replicated chromosomes, and this is pair of replicated chromosomes. These are sister chromatids, right? sister chromatids right here. We also call these 4 sister chromatids, we also call them a tetrad because there are 4. There are 4 molecules here. So this is a tetrad of sister chromatids, but these 2 are identical to each other, and these 2 are identical to each other. We can't say that these 2 are identical to each other. They're not. All right, so we swapped pieces, parts. Now let's draw the kinetochore. The kinetochore is it's a number of protein at the centromere. So technically speaking, that's the centromere. Actually, I wrote it in a place I don't want it to be. Okay, so this is the centromere, right? This is the centromere. It's this region. And the kinetochore is at the centromere. So the kinetochore comes here. Why do we need a kinetochore? Because the kinetochore is where the microtubule fibers that are emerging out of the centrioles is going to anchor. It's going to anchor that kinetochore and help pull towards it. So same thing happens here. Oh, I forget that I need the space. Okay, kinetochore at the centromere. And let's draw it in pink. Now remember, we have cohesins keeping the sister chromatids together. But we also have, let me do a highlight so it looks really different. We have these cohesion molecules that are keeping the pairs together. So when the pairs come to the metaphase, they're actually kind of glued together. Of course, this is not a strong glue, but they're still glued together. That's why we have this tug of war. But this time, instead of being the fibers coming from both sides of the kinetochore and pulling on the replicated chromosome and splitting the sister chromatids from each other. Here we don't have that. Here what we have is we have microtubules. Yes, they're coming only from one side. They're coming from one side. And then of course there's more like this that are going to other pairs. We have 23 pairs. I only drew one pair here. So this is going to be pulling towards the pole of the cell. So yes, we're going towards the pole. We're going towards the pole of the cell like this, pulling. But because we have these attachments here, this is keeping the pairs together. It's stuck there. So the microtubules from one end are saying, I want this one. And the microtubule on the other end saying, I'm pulling on this one, but they just won't come apart. Pull harder, pull harder. And eventually, ding, it's going to like separate from each other. And that's what happens in anaphase. Anaphase 1, the pair of homologous chromosomes finally separate. One chromosome of each pair moves towards the opposite pole, guided by the spindle apparatus. So this is metaphase, so we don't see the spindle apparatus. Let me see if I can show you a picture. Yeah, it's going to come up in the picture, so I'm not going to redraw this and make this lecture any longer than it already feels like it. So one chromosome of each pair moves towards the pole, but the sister chromatids remain attached at the centromere and move as one unit towards the pole. So these sister chromatids are moving together because they're still attached here. So they're going to be moving like this together. This is moving together towards this pole, and these sister chromatids are moving together towards the opposite pole. Okay, so they'll be split. They'll be split in the middle here. Boom, 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 boom. It'll be split. Oh, this is fun. All right, and then they will be pulled towards the pole. Okay, and then at telophase, telophase 1, each half of the cell has a haploid set of chromosomes, right? We only took one of chromosome 1, the original pair, one chromosome 1 in the other cell. So each cell gets a whole set, but they're still in a replicated form. So cytokinesis usually occurs simultaneously, and it really doesn't completely finishes because we jump right into prophase 2 right away. But no, but cytokinesis takes place, of course. What am I saying? Cytokinesis does take place because we're separating the cells from each other, but the reformation of the nucleus and the finalization of telophase doesn't necessarily have to go all the way to the end, because we have to dissolve the nucleus again to enter this prophase number 2 now. Okay, and there is no cell cycle, like we're not re-replicating the chromosome here. It's literally 2 cellular divisions in a row. There's no interphase or anything like that happening at the end of meiosis 1. So meiosis 1 separates pairs of chromosomes or homologous chromosomes. Okay, very important. This is what you need to remember. This is the whole purpose. This is what leads to diploidy. Okay, and here we can see the chromosome pairs are like hanging out together. Oh, we're saying goodbyes, we're exchanging presents and souvenirs, and then we line up at the equator of the cell during metaphase. Then we have the pairs of homologous chromosomes that are finally going to split from each other, and they're going to and they're guided on the rails of these microtubules. So cytokinesis, of course, has to take place at the end of meiosis 1, and the cleavage furrow is the name of that separation that's going to take place. The nucleus doesn't normally reconstitutes completely because division for meiosis 2 is already starting. So we'll have prophase 2, metaphase 2, telophase 2, and followed by cytokinesis. And that, meiosis 2, is exactly what happens in mitosis. Exactly. So you have a spindle of apparatus formed, so all these microtubules. Chromosomes move towards the metaphase plate at the end of a prophase 2. In metaphase 2, the sister chromatids are arranged in metaphase plate. So we're still talking about the same thing, except now the microtubules are going to come from both sides of these replicated chromosomes. And we're literally going to, the tug of war is going to be to end up separating those sister chromatids from each other. So they separate in anaphase 2. And in telophase and cytokinesis is when we literally are now finishing up the look of these cells by giving them a nucleus each and splitting them completely apart from each other. Separation of the cytoplasm, that's called cytokinesis. So at the end of meiosis, we actually have 4 daughter cells, each with a haploid set of unreplicated chromosomes. That's it. And each daughter cell is genetically distinct from the other and from the parent cell. So these are diagrams for meiosis 2. So what was drawn here? 3 chromosome types, but we don't have them in pairs. So here's chromosome 1, 2, 3. 1, 2, 3. So we have all the chromosome types, but we're not having them in pairs. So after interphase, the sister chromatids are held together by proteins called cohesins. The non-sister chromatids are broken at precisely corresponding positions. So, and a zipper-like structure called synaptomnemal complex holds the homologous together tightly. And that's what I drew earlier. Synaptomnemal complex. I didn't write it over here, but basically that's what I had drawn over here. Synaptomnemal complex. Where's my drawing? Here it is. So these big, see these big. So these are big attachments. So these big attachments are basically that's the glue between the two homologous replicated chromosomes. Okay, so the sister chromatids finally split. So just like mitosis, where we have the tug of war basically at those replicated chromosomes, and they're going to split. And now we call them individually, we call them chromosomes. We're done with using the term chromatids anymore. And then in telophase, we finish by wrapping up these chromosomes into nuclei, so a nucleus for each cell. And that's it. And I already said all that. So in meiosis 2, we separate the sister chromatids from each other. So when do we have the introduction of variation in these events? Well, during crossing over and synapsis of prophase 1. If these chromosome pairs were not exchanging genetic material because they're so sad and they want to keep a souvenir from each other, if they didn't do that, we would not have variation amongst us. We would literally all be identical. And how awful would that be? Because if you have a specific mutation that doesn't make you able to live with the sunlight, I mean, that's far-fetched, but why not? All of us would die in the sun. So, it wouldn't be good for the species for all of us to be clones of each other, because if something didn't work, then all of us would, be affected. All right, so this is just a diagram that shows you DNA breaks, the centromere, the cohesions that are, within the sister chromatids, and then the synaptomnemal complex between the homologous chromosomes, okay? So it's a slightly different way of attaching things. Okay, and what happens during crossing over? I think I went over that with the drawings. I used purple and green, and I literally kind of switched an arm and put it on the other side. So mitosis conserves the number of chromosome sets, producing cells that are genetically identical to the parent cell. We saw that in mitosis. Make sure you understood that. We have these chromosomes in sets. So 2 pairs of 1, 2 pairs of chromosome 2, 2 pairs of chromosome 3. Those are sets. Meiosis reduces the number of chromosome sets from 2 diploid, that's the diploidy we had, to only 1. That's the haploidy that we're looking to get, producing cells that are different genetically. So not only are they haploid, which is perfect genetically, we only need 23 because we're going to meet Miss Oocyte soon, and she provides 23, so it'll be 46. So in addition to needing only 23 chromosomes, our chromosomes are going to be different. And that's what these crossing overs create. They create different chromatids, different from each other, and it adds variation to these sperm cells and oocytes. So this is the overview of these 2 cellular division processes. You have to be familiar with that. This is very important. This will come up later on in your life. Plus, it's nice to understand that. Mitosis was 1 cell, and it gave rise to two daughter cells. And we went through all of these steps back in that chapter. So that parent cell starts the same way, 2n equals 6. So some of these parent cells are destined to make gametes, and some other parental cells are meant to only produce the cells of their tissue. So a skin cell will produce more skin cells, you know, if we needed more skin cells. So I'm pointing to that, but like let's assume this is a skin cell trying to reproduce. Okay. All right, so let me add one more thing. So we've said that the chiasma formation leads to the production of specific chromosome, a specific combination of genes, which will make all the chromatids different from each other. Okay. Now, so this is just a summary. I'm not going to read the summary. I do want to point out to you something. Look at these daughter cells. They are so different from each other. Look at this. Long chromosome 1 that's completely red. Long chromosome 1 that's completely blue. So this was the original maternal. This was the original paternal. Okay, so this daughter cell received the maternal version, and this daughter cell received the paternal version of that chromosome. Okay. And look at this, chromosome 2, chromosome 3, chromosome 3, chromosome 3, chromosome 3. Every single one of them is different. Chromosome 2, same thing. Every single one of them is different. However, how is it that we have this chromosome number 1 in this cell and this one in this cell, but we have this blue one in this cell and we have the red one in this cell? What happened? So this is what happened. So I'm going to skip the summary. Those summaries is for you to read after you study, if it helps you. So what's going to happen in anaphase 1 is very important for the additional contribution of variety. Hear me out. Synapsis and crossing over was very important to kind of create some mishmash of chromosomes, okay? And that kind of like adds variety to the chromosomes. However, the homologous pairs that are forming at the metaphase plate is very unique. The pairs of chromosomes, okay, and I'm not going to draw with crossing overs, but you see how we drew a pair of chromosomes like that, and then we said that, and then this chromosome is going to be pulled up, and this chromosome is going to be pulled down. Remember? Okay. And they were attached here, and then there was some tug of war, and then poof, they're going to separate. Okay. However, how they line up at the metaphase plate is random. So this was for chromosome 1, so this is number 1. But for chromosome number 2, hey, who knows? Maybe it decided to go here. So this is number 2. And number 2 was being pulled over here. So having the distribution of these chromosomes along the metaphysial, the metaphase plate is random. So you could have, from the pink set, you could have number 1 go up, and all the others, the pink set, is going to go down. So what's going to go up is the orange set, chromosome 2, chromosome 3, chromosome 4, blah, blah, blah. They're going up towards the pole of the cell that's at the top. And It just so happens that for chromosome 2, the chromosome 2 that's from the pink parental person that has the pink chromosomes, um, those are destined to a different pole than the original first one. So that is complete randomness. Okay. Separation of homologs during anaphase. Those are 3 events unique to meiosis. All 3 occur in meiosis 1. You need to know that. Okay. Sister chromatids cohesion allows the molecules, allows the molecules to stay together. We already saw that. Okay, I think I mentioned all of that already. So genetic variation produced in sexual life cycles contributes to evolution because we're constantly actually pushing to keep certain traits and get rid of other traits. Literally, it's amazing. For example, certain people in the classroom around you, certain people have much darker skin than others, uh, all the way to dark black skin. Why? Because evolutionary, these people adapted to a place where it's very sunny and it could contribute to skin cancer. And it's not good for your skin. So, um, so the people that lived in those world regions where there was so much sun and it could, in a detrimental way, um, evolved in being able to produce more melanin through their melanocytes. So melanocytes are skin cells that are next to the other kinds of skin cells, but those are just producing pigment. So all of my freckles here, those are the result of melanocytes producing brown pigment, because some of us are just born like that, and freckles is a trait that's passed on, and even dimples. So all of these freckles here, those are melanocytes producing melanin. And yes, sometimes it accumulates in freckles. So the hue of your skin is made of the color melanin and a couple other things, but melanin. So what's different between us and somebody with a darker skin is that the melanin activity is higher in those people. They actually don't have more cells, they just have a higher activity per cells. So these people have a lot of melanin production and end up with very dark skin. So genetic variation produced contributes to evolution. So how do we can create you know variation? Well, we can have mutations like spontaneous mutations, and literally that's going to create just another variation of the gene. So that can happen and happens. Mutation create different version of the gene called alleles, and we already talked about that. I think in a different chapter already, but I mentioned it in this chapter at the beginning. And reshuffling of the alleles during sexual reproduction produces genetic variation, which is that randomness that I talked about. That introduces genetic variation. Okay, so independent assortment of chromosomes, crossing over, and random fertilization. So what does that mean? Independent assortment, homologous pairs of chromosome orient randomly at the metaphase 1 of meiosis. So the pairs, they're like, oh, I don't know, let's go here, think, and they get stuck here, and then they're in this particular, orientation. And then another pair comes, and they're like not paying attention to where the other pair is and how it is and how it's likely. And it gets like stuck here in the metaphase, but the origin of these 2 chromosomes is actually kind of like opposite to the other 2 chromosomes. And that's it. So, all the parental chromosomes, well, it's just random. Let's just keep saying that. So, these pairs are like just lining up randomly on this metaphase. And when they finally split, the pairs split at the end of meiosis 1 or during anaphase 1, they're going to end up at separate poles. So that's called independent assortment. Each pair of chromosomes sorts maternal and paternal homologs into daughter cells independently of the other pair. So the number of combination possible when chromosomes assort independently is 2 to the exponent n. Even that, it's not quite accurate, but this is what we go by. 2 exponent n, where n is the haploid number. If you were to be haploid, you would have that number. So for humans, it's 23. So 2 exponent n, for us humans, that would mean 8 million possible combinations of chromosomes. So now you know why it was uh, uh, it added variations. So the diagrams coming up are showing you those possibilities. Either all the red ones line up facing the same pole, or they're facing opposite pole. So if they're facing the same pole, then all the blue chromosomes from that original set, they're actually all going to one cell, and all the red ones are going to another cell. So it's basically cloning yourself. Or if there is no mix up here, I mean, if there is a random assortment and you end up with some blue ones and some red ones in one cell, that's what you're going to end up with. And this is going to be very different from the parent. So then in the second round of meiosis, what you're doing is just splitting the replicated chromatids, and you end up with 4 different combinations of potential gametes. Okay, so these slides are basically just honing in on this variation. So independent assortment was the first one. Remember, independent assortment, crossing over, and random fertilization. Those are the origins of genetic variation among offsprings. All right, if you get that, this is the picture we're getting into right now. Is one we're explaining how variation is introduced. So one, by independent assortment, crossing over, which actually happens before independent assortment. That could have been first on the list. Where crossing over takes place, we talked about that. And this is just showing crossing over. And and random fertilization. Okay, I couldn't remember the last one. Okay, so this one doesn't have anything to do with the cell division per se. This one is more about which sperm is going to win the race, and every sperm is holding a different set of genes. And this is basically the luck of the draw. Is just going to be poof, one of these, and no, whatever the combination of, paternal or maternal chromosome is going to be in each one of these sperm cells is really random. And so there is randomness to which sperm is going to fertilize the egg. All right. All right, so these are all the diagrams. I really recommend that you you draw step by step if you prefer. Create your summaries. All right, I cannot run this because this is a PDF and not. Let me see if I can pull up. Okay, so this animation is not available right now for this recording, but that's okay. It's in the PowerPoint, not in the PDF file, in the PowerPoint. So you can click play. Literally, I think the play button is somewhere here. Click play and it will show you this animation. And additionally, there's the availability of separate animations that I also like to show in the classroom, but they're posted on Canvas or on the platform that we're using to post these lectures. Isn't this amazing? We can actually see these points of chiasma in real um, chromosome crossing over. Okay, random fertilization, we already talked about that. And we talked about evolutionary significance of this genetic variation. It's very important that we stay all very varied between each other. And that's it. That's the last slide. And this was a long lecture, so I'm appreciative of your patience and hopefully you got the idea. All right.